Hello students, in this video I'm going to give you a little bit of a review of basic genetics concepts so that you can do the exercise 11. If you understand genetics really well and you've just had genetics in your lecture, you might not need to watch all of this or you could skim through it. So this is a review of genetics concepts before you do exercise 11. So let me move to my PowerPoint presentation and we'll start here. Genetics tutorial is what we're going to be doing, and I want to be able to use my, not a laser pointer, I don't want that. I want to be able to use my pen. Okay, we're going to take a look at monohybrid crosses, sex-linked crosses, and dihybrid crosses. All of these crosses are in exercise 11. So, monohybrid crosses. Here is an example of a monohybrid cross, and it's an example that comes out of your Campbell biology textbook that Gregor Mendel, did with pea plants. He decided to look at crossing generations of plants and trying to sort out how traits are inherited. And he discovered a number of things. So if we take purple flowered pea plants and cross them with white flowered pea plants, and these are plants, the purple flowered plants are true breeding. They breed true. In other words, they are going to be purple, and then if they mate with themselves, they're going to make more purple, 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 purple. Breed true to purple. And he took these two parents, purple flowered plants and white flowered plants, and he mated them, and he found out that in the first generation, that's F1, first filial generation, all the plants have purple flowers. Oh, that was interesting. If you know anything about genetics, you probably are thinking purple must be dominant to white. If this trait is inherited as a single gene trait, which it is. And then he decided to mate the F1 by F1, the siblings, the first generation siblings. And he made a nice cross of F1 by F1. And he looked at the second generation. In the second generation, he found, this was sort of surprising, he found that 3/4 of the offspring had purple flowers and 1/4 of the offspring had white flowers. So the white kind of came back again. And this is Gregor Mendel, came up with a rule that we still use today to understand why this happened. Okay? So what he decided was there's two alleles for each trait. So each individual plant has two alleles or two genes for flower color. And and there's different versions of the genes or different alleles of the gene. Sometimes the version is dominant, that it masks the expression of the other, and sometimes the version is recessive. It can't be seen if there's only one copy of it. So if we start with our parents, if we go back to our cross that I just made, we would have purple, true breeding purple flowered parent. So this individual would be big P, crossed with a true breeding white flowering plant, little p. And I use the uppercase and lowercase designations intentionally. All of the first generation offspring were purple flowered, and so that probably stuck out to you as saying, purple must be dominant. We're going to use an uppercase letter to designate dominance here. And an individual who is a true breeding purple flowered plant will have 2 copies of the purple allele. Individuals who are true breeding white flowering plants will have 2 copies of the little p's, or the white alleles. And during the process of making gametes, now this purple flowered plant makes gametes, right? Eggs or sperm. What happens is the law of segregation. The 2 copies of the gene separate when you make gametes, so that 1 copy ends up in each gamete. So you either have a big P or a big P. If you're from the purple flowering plant, you can make gametes that are like big P, or every gamete's going to be a big P because that's all you have. If you're a white flowering plant and you're making sperm, now plants don't have swimming sperm, some do, but not peas. I'm just going to make them that way. All of your sperm would have little p's. Okay? And then in the first generation, an egg meets up with a sperm and inherits one copy of the big P and one copy of the little p, and is purple because purple is dominant to white. All of our F1 plants are what we call heterozygous because they have two different genes for the same trait, two different versions or two different alleles for the same trait. The parents in our generation here, in our cross here, are homozygous because they have, homo means same, two of the same alleles for the trait. Now in our cross, we crossed an F1 by an F1. In other words, we mated two individuals that looked purple but were heterozygous. And in the second generation, we found 3/4 of the offspring were purple. That means they must have had at least one big P, right? We don't really care about the other allele. They could have another big P or a little p. And 1/4 of the offspring were white. So they must have inherited the two little p's. We can draw a Punnett square to show this cross and how and the types of offspring we expect to get from this cross, and we should see this kind of phenotypic ratio that 3/4 of the offspring are purple and 1/4 of the offspring are expected to be white. So I'm going to do that on the next slide. A Punnett square for the P for the F1 by F1 cross with two heterozygotes. A Punnett square is a simple way to look at a genetic cross and identify the expected offspring from a cross. In this Punnett square, we're going to need a 4-box Punnett. And what we do on a Punnett square is on the outside of the box, we put the gametes that each parent can make. So let's say this is my male parent and this is my female parent. On this side, I would put the types of gametes on the left side here that my male parent can make. My male parent can either donate a big P or a little p. My female parent gives the egg, and she can donate either a big P or a little p. And then, by random chance, one egg meets up with one sperm, so we expect in equal proportions to get a combination of the two big P's, a big P, little p, or a little p, big P. I always put the dominant alleles first, or two little p's. Hey, let's think about this. 3 of those 4 boxes, or 3/4 of the offspring, get at least one big P, so they should be purple. 1 of the 4 boxes has 2 little p's, is homozygote for the 2 recessive alleles, and should be white. And so we can see a 3-to-1 or a 3/4 to 1/4 phenotypic ratio of purple to white. 3/4 of these offspring should be purple and 1/4 should be white. That's these guys down here, right? That's the phenotypic ratio. Sometimes we write it as 3 purple to 1 white. If you want to write it that way, I like to use fractions because it makes it easier for me. What is the genotypic ratio? Oh, the genotypic ratio is the actual genotypes, the genetic makeup. And there's 3 different genotypes that you could get. Okay, 1/4 of the offspring get the 2 big P's. Half the offspring get 1 big P and 1 little p, and 1/4 of the offspring can get the 2 little p's. That's what's expected, at least, right? It's not always exactly the offspring you produce because the meeting of the sperm and the egg are random. So it's not always perfect. Okay, that's a Punnett square for a monohybrid cross. We'll move on to sex-linked traits. Now, sometimes traits are inherited on autosomes, and sometimes they're inherited because of genes on sex chromosomes. Autosomes are all the chromosomes you have that are not the sex chromosomes. And for peas, that purple flowering trait was inherited on an autosome. The flower color was inherited on an autosome. But sometimes traits are carried, their genes are located on the X chromosome. Now, we have to think a little bit about, and these would be called sex-linked traits. We have to think a little bit about our genetic makeup. In humans, we have 46 chromosomes, or 23 pairs of chromosomes. 22 of these pairs are considered autosomes. They are the same in all individuals. We all have 2 copies of chromosome number 1. We all have 2 copies of chromosome number 2. We all have 2 copies of chromosome number 3, all the way up to chromosome number 22. And then our 23rd pair is a little bit different. Some of us inherit 2 X chromosomes, 2 long, big X chromosomes, and some of us inherit 1 X chromosome and 1 Y chromosome. That's what most individuals inherit. Sometimes we can get anomalies to that as well, which is kind of cool. Chromosomal females inherit 2 large X chromosomes. Chromosomal males inherit 1 large X chromosome and a Y chromosome. Okay, so females would inherit these 2 long X chromosomes, chromosomal females, I should say, and then chromosomal males will get 1 large X chromosome, oopsie, those should be attached, and a Y chromosome that's really small, okay? And sex-linked traits are traits whose genes are located on the X chromosome somewhere, not on the Y chromosome. And so males inherit just 1 X chromosome. So whatever gene they have on that X is what they express. They only get 1 copy of the trait, 1 copy of the gene. Females get 2 copies of the gene, and so they have kind of an expression that's very similar to autosomal expression. And so this is going to affect our inheritance patterns. And we have to, when we know a trait is sex-linked, it changes how we set up a Punnett square, and it changes the expected results of the phenotypic ratios in offspring, etc. I want to give you an example of that because we do this in our exercise 11 as well. So I'm giving you an example of a trait in humans, and we're going to talk about color blindness. There are actually several types of color blindness. I'm just going with generic right now. Many of these color blindness types are actually sex-linked traits. So in humans, let's talk about red-green color blindness. Is a sex-linked recessive trait. That means it's carried on the X chromosome. Its gene is somewhere on the X chromosome, and it's recessive. So you have to, so if you inherit 2 copies of the gene, they both have to be recessive in order to show color blindness. Suppose a colorblind woman mates with a male with normal vision. Okay, so we got this colorblind woman and a male with normal vision. And then I've got my problem. If they have a girl, what's the probability she will have color blindness? If they have a boy, what's the probability he will have color blindness? And I've got my symbols that I'm going to use. I'm going to use an uppercase B for normal vision and a lowercase b for color blindness. Now, remember that females are going to inherit 2 copies of this gene. Males are only going to inherit 1 copy on the Y chromosome. And so we're going to write this a little bit differently than we did with our autosomal traits. Let's solve this problem. So we've got this female who's colorblind. She's going to have to inherit 2 little b's. And these b's are on the X chromosome. So I'm going to write her genotype with the X's in it. And that's going to help me sort of keep track of the sex ratio as well as what's going on with our genetics. She's colorblind, so she inherits 2 recessive alleles that are both on the X chromosome. And she mates with a man who has normal vision. All right? A man has an X and a Y chromosome. And to have normal vision, he has to have a dominant allele for normal color vision. It's on the X chromosome. He doesn't have anything on his Y chromosome. He only gets the 1 copy of this gene, but it happens to have normal vision. Okay, now when we solve for what kind of offspring this family could have or is expected to have, we're going to keep our X's and Y's in the gametes. All right. When this female makes gametes, she can make the 2 X chromosomes segregate. All right. And so if I'm going to draw a Punnett square here, this female actually can only make 1 type of gamete. Her only type of gamete that she can make is an X with a little b. So we're going to put the eggs over here, the X with the little b. Or she can make an X with a little b. Or she can make an X with a little b. That's all she can make. I really don't even need a 4-box Punnett here. I could do this with a 2-box Punnett, but I guess I'll I'll do it with a 4-box Punnett. The male, when he makes gametes, the X and the Y chromosomes segregate. And so one sperm has an X and one sperm has a Y, right? So one of his gametes could be an X with a big B, and one of his gametes could make a Y. It could be a Y. And they should be made at about equal ratios. Now, when we look at the potential offspring from this cross, let's mix them together. Okay, this is going to be a female who has one big B and one little b. In other words, a female who has normal vision. Okay, our second box here, our second row here is similar. All of the females here have normal vision. And this box over here, the male, I know it's a male because it inherits a Y chromosome, inherits the recessive allele for color blindness. And this is an identical box. Oh boy. So it looks like all of the females from this cross will be phenotypically have normal color vision. And the males from this cross, all of them will be colorblind. Okay? So when I go back to answer my question, if they have a girl, what's the probability she will have color blindness? Well, there's 0. If they have a boy, what's the probability he will have color blindness? If it's a boy, we only have to look at the boy side, and we would say, oh, all of the boys are colorblind. Okay? If they have a child, what's the probability the child will be colorblind is 50%. Does that make sense? Because on average, 50% of the time you have a girl, 50% of the time you have a boy. 2 of the 4 boxes show color blindness. So the wording of the question actually is pretty important for solving genetics problems. Okay. So in sex-linked problems, the key is always to set up your genotypes so that the female has 2 X chromosomes with the genes on the X chromosome, and the male has 1 X chromosome and 1 Y chromosome. The gene is only on the X chromosome. This will help us when we set up problems for exercise 11. Okay, and now we're going to do a dihybrid cross. This is when we're dealing with 2 genes at the same time. What if we're studying 2 traits at the same time? How does that affect our what the plants inherit or what the offspring inherits? Well, we're going to take a look at flower color and plant height in pea plants. And we know some things. We know that purple is dominant to white. We saw that already back in our last monohybrid cross. We also know that tall is dominant to short. So short is going to have the designation little t, tall is going to have the designation big T. So if we start with true breeding parents, the first parent is purple and tall. The second parent is white and short. What are the genotypes of these parents? Well, okay, the parent that's purple and tall would have to have 2, you have to have 2 copies of each gene, right? Except if it's sex-linked. These are not. These are autosomal. And the purple tall parent then would have 2 big P's and 2 big T's, right? Two copies of each gene. The short white parent would have 2 little P's and 2 little T's. And each of these are homozygote for both traits. I would say the purple tall parent is homozygote dominant for both traits, and the white short parent is homozygote recessive for the white short traits, okay, for the 2 traits of flower color and height. Now it says that the first generation offspring are all purple and tall. That's no surprise because we know purple and tall are dominant. But what genes do they inherit? Well, remember, when this plant makes gametes, and this is called the law of segregation, but actually it isn't. When this plant makes gametes, there should be 1 copy of each gene in each egg. In other words, there should be a P and a T. One P and 1 T. Well, this one can only make 1 type of egg, the big P and the big T. That's the only kind of gamete that parent can make. This guy over here can really only make 1 type of gamete too. It has 1 copy of the P, 1 copy of the T. It's just the recessive copies. Okay? And then an egg and a sperm meet up and make an F1 offspring. And all of the F1 offspring should have the same genotypes. Let's put the P's together and the T's together. All of the F1 offspring should be heterozygote for flower color and heterozygote for height. But because purple and tall are dominant, all of them should be purple and tall. We would actually call this a dihybrid plant. It's diheterozygous. It's heterozygous at both genes. Okay, now what if I decided to mate my F1 plants together? What if I decided, let's cross our F1 plants and our siblings and see what kind of offspring we get in the next generation? That's when we get into fun. Let's cross the F1 by F1 plants. This is called the law. This is going to show you the law of independent assortment. And let's think about what kind of gametes the parents, so these are our F1 parents, right? And we're thinking about what kind of F2 plants are we going to make down here, right? We first have to think about what kind of gametes can each parent make? What kind of eggs can this parent make? What kind of sperm can this parent make? Remember, in the formation of gametes, each gene separates so that there's one copy of each gene in each gamete. And it turns out that the color gene is independent from the height gene. In other words, let's think about this. This parent could make a gamete with the big P or a gamete with the big T. Okay, a gamete with a big P and the big T. That's one type of gamete it could make, because it has to have one copy of each gene in each gamete. It could put the big P with the little t. It's possible. It could put the little p with the big T, or it could put the little p with the little t. And these are all equally likely. About a quarter of the gametes should be big P, big T. About a quarter of the gametes should be big P, little t. About a quarter of the gametes should have the little p and the big T, and about a quarter of the gametes should finally have the little p and the little t. This guy making sperm can make 4 different types of sperm as well, and they all should be equally likely. So let's draw those in. Big P, big T, big P, little t, little p, big T, or little p, little t. And now, when one sperm meets up with one egg, this is a random process. So think about all the different combinations you could get. You could get this egg meeting up with this sperm, or this egg meeting up with this sperm, or this egg meeting up with, hey, let's make a Punnett square. And we will look at the potential offspring. So I'm going to make a giant Punnett square here. It's big, so that's why I'm trying to use as much of the board as I can. I'm going to make it it's going to have 4 boxes on each side, because there are 4 different types of gametes that each parent can make, right? The parent 1, the female, can make a big P, big T, or a big P, little t, a little p, big T. Remember, there's always one copy of each gene in each gamete. So there has to be a P gene, there has to be a T gene, but only one of them. You can't put 2 P's together or 2 T's together, or you can't just put a P and a T separate. Otherwise the poor sperm would be missing a gene for a whole trait, right? That would be terrible. Okay, so always one copy of each gene. Okay, now I can fill in this giant punnet, right? Big P, and I'm going to put the P's together. You fill this all in, guys. It's going to take a while. And I'm terrible at doing Punnett squares that are this big. I'd rather use the rules of probability. So if you're learning those in your lecture, that would be fantastic, because I'm so bad at this. I always make errors. Okay. I know it's hard to keep them all straight, so we're going to see a pretty complicated picture here, but when we look at the phenotypic ratio, the types of offspring that we could get and their ratios, it's not that complicated. And in fact, you'll recognize the dihybrid cross phenotypic ratio is what we'll call it. Okay. I'm working hard here. P, P, T, T. Whew, this is hard, guys. P, P, T, T. Okay. If you look at that and you sort it all out, you will find that 9 of these 16 boxes, or 9/16 of these, will have the gene for purple and tall. They'll be dominant for both traits. So like 9/16 will be both purple and tall. I'm just going to put like a dash here because I don't really care what the second gene is for each of these traits. I do know that 9 of the 16th will have the trait for big P and big T. Like this one is one of them, you know? Okay, and here's another one that's one of them. And there's 9 of them. 3 of these boxes out of 16 will have the will have the dominant allele for purple, but then have 2 recessive alleles for short. 3 of, let me see if I can find one of those. Here's one right here. Okay, there's 3 of those suckers. 3 of the 16 boxes will have the 2 recessive alleles for white, but the dominant allele for tall, and be white and tall. And 1 of the boxes, this little guy down here, will be both white and short. Will inherit the homozygous recessive condition. 9:3 to 3:1. This is the phenotypic ratio for a typical dihybrid cross. Yes, between 2 diheterozygotes. Okay? This is something that you should know and know how to do with a Punnett square as well. It's kind of tricky, but once you see it, once you got it. We're going to do a dihybrid cross with our Fly Lab simulation in exercise 11 as well. So that's it, and this is the review. So that's all I have for you. I'm going to get out of here if I can. And finish this up. Thanks.